Okay, so I think I'm emceeing our 7 minutes of science today, and it's only 7 minutes, but I'll take any minute from Abhishek. It is my pleasure to introduce today Abhishek Srivastava, who came to us in 2019, just ready for the pandemic. Where is he? Oh, there you are. He got his PhD from Wisconsin with Mark McBride, and then had a postdoc at Harvard with one of my heroes, like Howard Berg. And he combines, that's why we were so excited to have mechanistic understandings of microbiology and brings them to bear into microbiome science. And so we were hoping that having him here would help us dislodge microbiomics from the correlational basis, and he's been doing just that. So, Abhishek.
Thanks for the very kind introduction, Farhan. As Farhan mentioned, I work with bacteria, okay, and up until about 30 or 40 years ago, people used to think that bacteria are just a bag of enzymes, okay, and which has certain kind of membrane, and then just enzymes float around and they diffuse. Okay, but now we know through the work of several very talented and hardworking people that that is not true, and that bacterial cells indeed have some sort of a structural organization, and they have very well-defined macromolecular assemblies. Okay, now some of these macromolecular assemblies are called as bacterial protein secretion machineries, and their job is to secrete proteins outside the cytoplasm into the extracellular space of the bacteria. Okay, and they have the various functions. Some of these proteins can be enzymes which break down carbohydrates. Some of these can lead to pathogenicity. Some of these can be important for intracellular competition and cell adhesion, biofilm formation, motility, etc., etc. My lab focuses on one very specific type of protein secretion machinery, which is called as the type 9 secretion system, or is abbreviated as the T9SS. Okay. Now, the type 9 secretion system was discovered only about 12 years ago, in fact, in the lab of my PhD advisor, Mark McBride. And at that time, I did some of the very early characterization of the core T9SS protein, and seems like I enjoyed it so much that I'm still working on this machinery even today. And our main interest was studying how this machinery controls motility and biofilm formation at the time. But very recently, for example, this paper came out just 2 weeks ago. It was shown that the type 9 secretion system of a gut bacterium is involved in the secretion of a protein that sequesters trypsin, which is a protein degrading enzyme in the gut. Okay, and it breaks down this trypsin such that this trypsin is not able to break down immunoglobulins in the body, and so it boosts the immune system of the host. Okay, and these people over here, they also showed that when they took samples from patients that had diarrhea due to coronavirus infection, they found that this particular enzyme, which is secreted by T9SS, was actually upregulated. Okay, there are several other examples also. For example, over here, there's a bacterium in the human mouth which secretes certain enzymes that break down tissue, and it causes a disease called as periodontitis. But that enzymes that breaks down the tissue is called as gingipain and is secreted by the type 9 secretion system. Okay. So about 2 years ago, it was discovered that this enzyme secreted by T9SS actually can be found in the brain of patients with Alzheimer's. Okay. And these were postmortem brain samples, and this particular enzyme secreted by T9SS was thought to be the precursor of beta amyloid in the brain at that time. Okay. But also, it's so T9SS is involved in secretion of enzymes that break down cellulose. When we eat salad, our human body is not able to break down the dietary fibers found in salad, but certain enzymes secreted by T9SS are able to break down the dietary fibers. So it's a very versatile protein secretion machinery, and it's found in a group of bacteria called as Bacteroides, which are found, which are very important members of the human gut and the oral microbiota. Okay, so in the past few years, this field has gained a lot more health-related significance, And as Farhan mentioned, I started my lab in 2019. Right now, we have 3 postdocs, 4 PhD students, and 5 undergrads. And we like to have fun, both in the lab, apparently, and outside the lab as well. And we are funded by NIH and IGMS, NIH and IDCR, and by CSA to do some of the research that I'll tell you about today. Okay. So before I start telling you about the work of my current lab, what we know about this type 9 secretion system machinery is that it has the ability to move proteins on the surface of a cell. So here you see there's a video where I attached a gold nanoparticle to a protein that moves on this, that is located on the cell surface of this bacterium. And this protein actually moves in a conveyor belt-like fashion. And recently, the protein component of that conveyor belt was also discovered. And we were wondering back in the day as to what is the driver of this motion. Okay, what causes this motion along from one pole to another? And so, what we found is that if we break down the cells and we tether them on glass, these cells actually rotate around a fixed axis. So this told us that there's a rotary motor involved in this motion over here. Okay, and we later found that this rotary motor is actually driven by proton motive force. And around that time, we only knew of 2 PMF-driven rotary motors. One was the ADP synthase, another was the flagellar motor. And in that work excited a lot of structural biologists. And recently, this work came out from UT Health in Houston, where they were actually able to image the entire type 9 secretion system and some of its rotary component. And this is work from Oxford University, where they were characterized the stator complex or the PMF driving proton channels over here. And they came up with some sort of a simulation of how this rotation is actually happening. So we have a hypothesis that this rotary motor actually pushes a conveyor belt on the cell surface, which leads to motion of cell surface proteins, and thereby it leads to a linear motion of the bacterial cell. Okay. So I'll give you some snippets of the work that is going on in my lab so that you have a brief overview of our research directions. So this is work done by a third-year postdoc in my lab, Trivedi, and he's supported by a second-year PhD student, Emma. And as I told you earlier, this type 9 secretion system motor is a rotary motor. And in wild type, this motor is exclusively or is mostly counterclockwise. Okay. But what Trivedi did was he mutated a small component of this conveyor belt over here, and he finds that now this motor is exclusively clockwise. Okay. So he has changed the rotational direction of the motor, and by doing that, he has discovered the switch that controls the directionality of this motor. Okay. Now, so mostly in bacterial systems, such kind of a switch is encoded, is a sensory transduction protein which senses something, some sort of a chemical. Okay, but here this switch is in this conveyor belt protein. Okay, so we think that this conveyor belt is somehow sensing some sort of a mechanical stimulus, and that is leading to this change in direction of the motor over here. Okay, now this work done by, spearheaded by another postdoc in my lab, Gosai, shows that. Well, what Gosai discovered was that we do have a sensory transduction protein in these bacteria, and he mutated. He made a point mutant in that novel protein that he discovered, and he found that after that mutation, his wild type cells have the ability to swarm very well or move very well, but his mutant doesn't move that well. Okay, and his mutant also increases biofilm formation. So it seems like there are 2 levels of feedback in these type of bacteria. One is a mechanical stimulus, another one is a chemical stimulus. Okay, so we have 2 projects, 2-pronged approach, and we are kind of teasing apart both how the mechanical stimulus is being sensed by bacterium and how this chemical stimulus is being sensed by this bacterium as well. Now, as I mentioned, bacteria containing this machinery are also found in the human gut and the oral microbiota. And this work is done by Sophia, who is now a first-year PhD student, but she was, an undergrad researcher in my lab, and she apparently liked her undergrad research so much that she continued as a PhD student. But this is the work that she did during the final year of her undergrad. And she wrote a Python script and wrote a web scrape that actually collects data from a database called as the Human Oral Microbiome Database. And she studied what are the evolutionary relationships between motility machineries in the human oral microbiota. Okay. And we find that, okay, a small percentage of bacteria in the human mouth have the ability to move. But what we also know is that these motile bacteria, which are called as capnocytophaga, can carry other non-motile bacteria in the human mouth as cargo. Okay, so they can shape the spatial structure of a human oral microbial community. And this final slide just highlights the work of a second year PhD student in my lab, Nick. And so Nick came up with this idea that, okay, you've shown us that these non-modal bacteria can be transported as cargo, but can these swarms of modal bacteria transport other things like phages, which are viruses that infect bacteria as cargo as well? Okay. So he got some fluorescent bacteriophages, which you see over here, and he found that these bacteriophages do get transported by these modal swarms. And when he spots these bacteriophages, next to a colony of an E. coli cell, he finds that when the phages are being transported, the rate by which this colony gets disrupted is around 10 times faster when the phages are just simply diffusing. Okay? So these modal bacteria, not only do they carry non-modal microbes as cargo and shape the structure of a microbial community, they can also carry virus particles and can actually change how the microbiota would look like if you were to take it out and image it in real time. Okay. So that's the end of my story for today. This is a brief overview, but if you have more specific questions, please ask me later or at the end of this. Thank you very much. I think John has one over there in the back.
Where does that protein ribbon on the outside? So Bacteroidetes is a gram negative.
That's true.
So where does it attach and how is it sitting? I can't even imagine.
That's true. So it is in the outer membrane, but it's not. So it's not. So we have a track and then we have a thread. And peptidoglycan is the rigid structure on the bacterial gram-negative bacterial cell. So the track is attached to the peptidoglycan by some PBP, which is a peptidoglycan protein, binding protein. But the thread, which is the mobile component, slides along that track. That's the current model. And that's on the inner side of the outer membrane, essentially. And then the adhesion protein sort of stands out, and we can image that via EM. Yes, that is true. Yeah. But that track per se is in the outer membrane, essentially. But it's anchored by the peptidoglycan, so it passes through the periplasm to reach the peptidoglycan.
Okay. We'll have to.
Yeah.
We can talk about it later.
Sure.
That's amazing. Yeah.
Thanks.
So Wade Van Horn's stuff looks at the trip proteins and they have both sort of, if you will, mechanical sensing, which measures heat, and chemical sensing, which, measures like caspacin. So is this the same kind of situation where the same protein both senses mechanical and chemical things, or are
No,
they different?
they are different proteins. So,
Oh,
I mean,
okay.
I obviously didn't have the time to go into the details, but the second protein that I talked about is a totally different protein.
Okay.
It's not the conveyor belt protein, and it's mostly a cytoplasmic protein that senses some sort of a chemical.
But impacts the same system.
Impacts the same system, impacts motility goes down, biofilm goes up.
I got it.
That's what we see.
Right.
I think, yeah. Okay. It's why do you ask? Maybe I want to keep that as a mystery, but it's on the third floor of Biodesign. Yeah.
The spinning that you showed.
Yeah.
Is it energy dependent?
It is proton motive force dependent. Yeah. PMF dependent. Yeah. Thank you. No, it does not use ATP. It's the proton gradient, which flows to ion channels. So the ion channels move like that.
Do you notice any trends in which particles that it conveys out of the cell?
So as in it's moving along.
Well, you said that some of the proteins were Alzheimer's related,
Yes,
some of
that's true.
them were
Yeah.
other types of things, some were good, some were bad.
Yeah.
Is there any type of trend as to what they select with this chemo selection?
Yeah. So the secretion, there are two signals that we know of for secretion, which are on the C terminal of the secreted protein. So if we do bioinformatics, we can tell, okay, these are the group of proteins that are going to be secreted by type 9 secretion system. Okay. The type of proteins that we find associated with motility have a slightly different secretion signal, but it's still, we are still trying to tease that apart completely. In certain bacteria, there are actually this type 9 secretion system is more complex because you look at the structure, there are multiple types of pores on the outside ring of the type 9 secretion system. So
So
some
it's
pores,
stereoselective?
we don't know about the stereoselectivity at this point, but there are certain signals, protein signals at least, that what we know of are leading to selectivity, and some enzymes would be then soluble, some would be outer membrane associated, depending on those signals.
All right, last shot. Any other questions? All right, well, thank you. That was great.